Hi guys. Do you know what does DNA repair mean? DNA repair refers to a collection of processes by which a cell identifies and corrects damage to the DNA molecules that encode its genome. Many of these lesions cause structural damage to the DNA molecule and can alter or eliminate the cell's ability to transcribe the gene that the affected DNA encodes. Other lesions induce potentially harmful mutations in the cell's genome, which affect the survival of its daughter cells after it undergoes mitosis. Source of damage is divided into two, which is endogenous and exogenous. Endogenous is a reactive oxygen species produced from the normal metabolism byproducts, spontaneous mutation, especially the process of oxidation deamination. While exogenous was caused by external agents such as radiation, ultraviolet ray, toxin, mutagenic chemicals, viruses, and thermal disruption. Photoreaction, light repair. UV light strikes one of the adjacent thymines, creating a thymine dimer. DNA photolysis recognize the kink in the DNA, and bind to the site. When excited by blue light, 345 to 400 nanometers wavelength, the photolysis change conformation, breaking apart the dimer.
Excision repair, dark repair. A thymine dimer distorts the DNA molecule. A nuclease enzyme cuts the damaged DNA strand at 2 points and the damaged section is removed. Repair synthesis by a DNA polymerase fills in the missing nucleotides. DNA ligase seals the free end of the new DNA to the old DNA, making the strand complete. Mismatch repair. The DNA mismatch repair pathway plays an essential role in the correction of replication errors such as base-base mismatches and insertion-deletion loops that result from DNA polymerase misincorporation of nucleotides and template slippage, respectively. Mispairs generated by the spontaneous deamination of 5-methylcytosine and heteroduplexes formed following genetic recombination are also corrected via MMR.
First, a protein complex, group of proteins, recognizes and binds to the mispaired base. A second complex cuts the DNA near the mismatch, and more enzymes chop out the incorrect nucleotide and a surrounding patch of DNA. A DNA polymerase then replaces the missing section with correct nucleotides, and an enzyme called a DNA ligase seals the gap. One thing you may wonder is how the proteins involved in DNA repair can tell who's right during mismatch repair. That is, when two bases are mispaired, like the G and T in the drawing above, which of the two should be removed and replaced. In bacteria, original and newly made strands of DNA can be told apart by a feature called methylation state. An old DNA strand will have methyl CH3 groups attached to some of its bases, while a newly made DNA strand will not yet have gotten its methyl group. In eukaryotes, the processes that allow the original strand to be identified in mismatch repair involve recognition of nicks, single-stranded breaks, that are found only in the newly synthesized DNA.
Problem in DNA repair. How can errors occur in DNA repair? Error is a natural part of DNA repair. Nucleotide bases exist in two different chemical forms. One is common form and another one is rare form. Rare base pairing can happen if one nucleotide and base pair is the rare form instead the common form. This error caused by different but non-tautomeric chemical form of bases, which is bases with an extra proton, or normal bases that nonetheless bond appropriately. It was due to a slight shift in nucleotide's position, which known as wobble. Strand slippage during DNA repair. There are two situations for this problem. Firstly, slippage occurred during the repeated sequences and synthesized strand. One nucleotide was added. As a result, new strand has an extra nucleotide form. Secondly, slippage occurred during the repeated sequences and template strand. One nucleotide was leave out. As a result, new strand with missing a nucleotide form.